Genetics once again affirms creation, and we interview geneticist Dr. Jeffrey Tomkins. Again, this is Genesis Week. And a grand old welcome to this episode of Genesis Week, the weekly program of creationary commentary on news, views, and events pertaining to the Origins controversy, made possible by you, the supporters of Core Ottawa, Citizens for Origins Research and Education. Excellence in pirate broadcasting. We continue to bring you the information the anti-creationists don't want you to see or hear, and we give glory to our Creator while doing it. Remember, if you get lost in cyberspace, you can just head on over to Wazooloo.com or GenesisWeek.com, which takes you to our YouTube channel, where you can subscribe to get extras like Creo, Rants, and full interviews with our guests. I'm your host, Ian Juby. Genetics provides some of the most powerful evidence for creation, and partly because it is so incredibly complicated. But that means it's a challenge to present genetics in layman's terms so the average person can grasp it. So, before we turn to our special guest for today, I wanted to take a moment to give the viewers a crash course in genetics so you can follow along somewhat in the conversation with which we're about to engage. Now, genetics is a study of our genetic code, our DNA. The genetic code is essentially the plans or instructions on how to build you, or a plant, or an insect, bird, animal, etc. The code is very much akin to a computer program. Now, our computer programs use ones and zeros. DNA has 4 different characters it uses to write its programs. Now, while the computer stores, reads, and writes its programs and data using electricity, DNA uses chemicals to store, read, and write the program and data. The chemicals are adenine, cytosine, guanine, and thymine. And we will represent those chemicals by their first letters, A, C, G, and T. Now, A will only bond with T, and C will only bond with G. These bases, or base pairs as they are called, make up the middle of the DNA strand, which is like a twisted ladder. So to read the DNA is split down the middle, and the molecular machines can read one side, one character at a time. Because the other side of the ladder is a complementary image, that side can be read as well. A gene is a section of the DNA which is read and can do a number of things. It can be used as the plan to build proteins, which are the building blocks of every living thing. The gene can turn on or off other genes in the DNA, instructing the molecular machines where to go to read the DNA. They can activate or turn off certain functions within the body. Now, because of its importance, the DNA is heavily protected within the cell and doesn't leave the nucleus. So how then is the information transmitted and used outside of the cell nucleus? Well, it is used by making a copy of the program, which basically takes the form of one half of the ladder. This is called RNA. Copying the code onto an RNA strand is called transcribing or transcription, and the machine that copies the DNA is called a polymerase. Once the RNA is made, it can travel to all kinds of places where it is read. RNA that provides the assembly instructions for proteins are called coding RNAs. RNAs that provide special coded instructions and are not used in directly building proteins are called non-coding RNAs. And of course, some RNA is longer than others, so you have short non-coding RNA and long non-coding RNA. In the DNA, you have introns and exons. Exons are segments of the DNA that are copied into coding RNAs, which ultimately make proteins. Introns are segments of the DNA that are copied into non-coding RNAs, which have, of course, a wide variety of purposes. All of the genes in the DNA are marked by a start and stop sequence of the letters. These sequences are called codons. Start codons mark where to start reading the DNA for a specific gene. The stop codon tells the molecular machines where the gene ends. Also involved in the process of transcribing are promoter regions. The molecular machine that reads the DNA must attach to the DNA. It looks for a specific promoter region which indicates which gene it is about to read. If it's a match, it locks onto the DNA, looks for the start codon, and starts making a copy. You can think of the promoter region as almost like an address. Now this will become quite important in a minute because if you do not have the promoter regions, it's like trying to find a house without an address. The DNA ladder is twisted up tight and then wound tightly onto spools and then into bundles called chromosomes. Now these chromosomes are big enough to see under a microscope. All the DNA in your body could fit in a teaspoon. Yet, if you were to untwist and unwind all the DNA in your body and stretch it out end to end, you'd die. But your DNA joined end to end would reach to the moon and back some 1,500 times. That's some pretty impressive programming. Microsoft could take some lessons from there. Now, the great apes have 24 pairs of chromosomes, but humans only have 23 pairs. But it is assumed that humans descended from the great apes. It was claimed then that two chromosomes fused together into one at some time in the past as humans were evolving. It was believed that human chromosome number 2 was the resultant chromosome composed of two fused ape chromosomes. We discussed this claim previously on several shows, as well as Creo Rant number 181, and how the chromosomal fusion argument is bust. More news has come out since the airing of these shows and our previous interview with Dr. Jeffrey Tomkins, who joins me again. Dr. Tomkins' PhD is in genetics with a master's in plant science and a bachelor's in agriculture education, and he was on faculty in the Department of Genetics and Biochemistry at Clemson University for a decade. He also ran his own genomics institute for 5 years. He is now a research scientist with the Institute for Creation Research, where he continues to publish various papers. Some of those papers we're going to discuss today. He joins me from his office in Dallas. Welcome to the show again, Dr. Tomkins.
Thank you. Good to be back.
Now let's start off. I want to backtrack to a paper you came out with in 2011 addressing this claim that humans have a fused chromosome, which allegedly supports evolutionary claims.
Right.
So let's go back in time for a minute, back to 2006, and revisit a lecture given by evolutionist Dr. Kenneth Miller at Case Western University. The lecture is entitled The Collapse of Intelligent Design. Now, it's interesting to note he very confidently claims that not only is there evidence that humans had two chromosomes fused, but that this is such powerful proof of evolution that he brought it up in the Kitzmiller-Dover trial. A court case which frankly wasn't a court case, but a three-ring circus complete with a circus judge. I mean, a circuit judge. The segment of Miller's lecture where he discusses the chromosome fusion was linked to all over the internet. In fact, even today, I get comments from atheists and skeptics pointing me to that segment posted on YouTube under the provocative title, How to Shut Up Pesky Creationists. They hand me this video as if I haven't seen it before, as if it's absolute proof of evolution. I'd like to play a segment of that lecture so we can hear straight from the horse's mouth exactly what Miller said.
What must have happened is that one pair of chromosomes must have gotten fused. So we should be able to look at our genome and discover that one of our chromosomes resulted from the fusion of two primate chromosomes. So we should be able to look around our genome. And you know what? If we don't find it, evolution is wrong. We don't share a common ancestor.
Okay, so in 2011, you and Dr. Jerry Bergman came out with a paper showing that this alleged fusion site was small, highly degenerate, and really not much of a fusion signature.
Right.
2 years later, you published a second paper showing that the fusion site actually contained the genetic code for an important feature inside a highly expressed, life-supporting gene. Now, we discussed this when you were on the show last, and I got a firestorm of emails and comments saying that you were wrong, and even Kevin Miller said you were wrong. So clearly you were wrong.
Okay, well, should we backtrack and...
Sure.
and cover the details of that discovery? Okay. Well, the fusion site actually is a functional feature inside an RNA helicase long non-coding RNA gene. So this gene produces functional long non-coding RNAs of various sizes. Now this gene has 3 exons, or 3 coding regions. The other areas of the messenger RNAs are spliced out of the transcripts made from this gene. So you get short variants and you get long variants of this gene. So Ken Miller actually gave an interview to a reporter that works for, what was that magazine? I've got it here, Mother Jones.
Oh, yes, yes.
Magazine, okay. Does that sound familiar?
Yes,
okay.
It does, yes.
So the reporter was a guy named Mooney. So Mooney basically got the interview from Ken Miller. So Ken Miller says that the fusion site is actually 1,300 bases away from the gene.
Right.
So a friend of mine at the Discovery Institute said, Jeff, did you see this article? And I said, no, I didn't. He said, well, this is what Miller is saying. He said, you need to contact him and get this thing settled. He said, you know, Ken Miller never responds to me whenever I send him an email, but maybe he'll respond to you. So I sent Ken Miller an email, and he actually responded within an hour. Very quick. And he was very cordial. He gave a nice long email explaining his position. But the interesting thing was he obviously didn't read my paper, because if he would have read my paper in its entirety, he wouldn't have said the things that he said. So I basically had to explain my entire paper to him over a course of several emails. And then he finally realized that he had made a mistake and that I was right. And so he said in the last email, talking about the long transcript, not the short transcript, or the long messenger RNA produced from this gene that has all 3 exons, he says, in this transcript, the fusion site is in the middle of the first exon, as you know. Well, actually, it's not in the middle of the first exon, it's in the middle of the first intron. But anyways, he acknowledged I was right. And then Casey Luskin actually took this piece from this email where Miller acknowledged I was right and published it in an article in The Blaze, which is an online media operation. And so people can actually get this article that Casey Luskin published with Ken Miller's quote from the email admitting I was right. So basically, everything is exactly as I described it, but not as I'm describing it really, but it's because that's what the data says. Yeah, one thing that's interesting about the fusion site, when they first discovered it back in 1992, now when I talk about when they first discovered the DNA evidence for it, when they sequenced what's called a DNA clone from human chromosome 2 from that region, when they found these telomere repeats, even though they were degenerate, it was not what they were looking for. They say that in the paper because they know that when fusions occur in nature, in animals that actually survive these things and sometimes even reproduce afterwards, that these fusion events always involve something called satellite DNA. So you can have, actually the PowerPoint presentation I gave you has got a picture describing it. So you can have a chromosome break and have 2 regions of satellite DNA fused to each other. You can have a telomere fused to satellite DNA, but you never see telomeres fusing to each other. In living animals, sometimes you will get telomeres that will fuse to each other in cancer cell lines, which is, I think it's safe to say that's not a healthy
No, definitely not.
living tissue, but I have evolutionists always say this. Yeah, well, look at cancer cell lines. Telomeres fuse in there. Well, that's not a healthy living tissue indicative of a healthy organism. Cancer genomes are completely rearranged and completely haywire.
Right, definitely not onwards, upwards
Because
evolution.
No, they're not. They don't represent,
Right.
functional normal cells. So anyways, we never see these things in living animals. So when they discovered this degenerate telomere region, it was not what they were looking for. They were looking for something that had satellite DNA in it. So if the fusion, if the human chromosome fusion was real, it would be the first documented instance of a telomere-telomere fusion ever occurring in nature.
Right.
But obviously that's not what it is.
Now, you just came out with a paper in Journal of Creation you co-authored with Brian Thomas, also there at ICR with you, with the provocative title, How Reliable Are Genomes from Ancient DNA? Now, this is an excellent question. You deal with the DNA that was found in what is claimed to be extremely old fossil skeletons of animals, and in particular humans. The Denisovan skeletons were one I've discussed here on the show before. Why don't you share a little bit about your paper, and why this question is so significant? Why should we care about how reliable the genomes of ancient DNA are?
Okay, well, there's actually a story behind this, that sort of my own personal story and quest is behind this. I began downloading these ancient DNA sequences from Neanderthal, from Denisovan, and I began noticing that there were some serious quality issues with these sequences. And so I wanted to really get to the bottom of what was going on. So I contacted a researcher at the Max Planck Institute, which is kind of the center of the universe for ancient human DNA, or what they call archaic human DNA now. By the way, that's what they officially call the Denisovan and the Neanderthal specimens, is archaic humans now. That's what the people in the field call them. Yet there's all this controversy. Are these humans? Are these not humans? Blah, blah, blah. Well, the people in the field are calling them humans. So anyhow, so I began actually corresponding with this researcher at the Max Planck Institute. They were very nice, very, very cordial. They gave me access actually to not only raw sequence, but also their new Neanderthal genome assembly they had not yet published.
Oh, wow. Okay.
Which I thought was very nice. And so I started by downloading the smaller chromosomes, the Y chromosome and chromosome number 22, because my computational resources at that time were not as powerful as they are now. Now we've got some very nice computational servers I have access to, and I can handle very large data sets. But at the time, I didn't have that. So I downloaded these sequences and began working with these assemblies. Now, these are what are called assemblies, where they actually took the raw Neanderthal DNA sequence, they cleaned it up, and then they assembled it into alleged chromosomes representing these ancient humans. And so I began working with these sequences, and basically, overall, my assessment was that the quality wasn't that great, but it was basically just human DNA for the most part. And then I began emailing back and forth, and then what the researcher that I was working with said, well, quit working with the Y chromosome when now we think that this is a female individual.
And only males have Y chromosomes.
And so I'm thinking, well, what's this Y chromosome doing in here? Now you think it's a female? Well, it turns out that this is not a hidden controversy. It was actually discussed in several journal papers. And their excuse was, well, we found a few genes that were similar to those on the X chromosome and accidentally misassembled this entire Y chromosome, based on these sequences they're called X degenerate genes. that are on the Y chromosome. But anyways, they claim they got their X and Y chromosome sequences mixed up. Well, that really is a pretty poor excuse for assembling an entire Y chromosome and then claiming it all of a sudden it's a female and then getting rid of it. So at that point, I lost all faith in the Neanderthal sequence and the Denisovan sequence and quit working with it. And I also began studying the issue, and my co-author Brian Thomas did a lot of study on DNA kinetics and how fast DNA actually deteriorates under these types of conditions. They're finding these skeletons and these bones. And really, there really shouldn't be much DNA there to begin with, because DNA deteriorates very rapidly in just a few thousand years. Yet they're claiming, I've got the data here, they're claiming in some of these cases that these individuals are 50,000 to 300,000 years old, which is really absurd. There shouldn't be hardly any DNA there after 5 to 10,000 years. And of course, that fits with a biblical time frame. And based on studies and how fast DNA decomposes and falls apart, it's totally in line with a biblical timeline of just 5 to roughly about 5,000 years. I think these individuals were probably from after the flood. The DNA is probably 3,000 to 4,000 years old or so. I think these were individuals after the flood that were forced to live in caves and things as civilization began to be built back up. They're not found in flood sediments. They're actually buried skeletons that people died and then their relatives buried them, basically, is what it looks like for the most part. So when I began studying this issue in even more detail, I realized that the amount of modern human DNA contamination in these samples is really high and a really big problem. So that Y chromosome that was in that assembly was probably from modern humans that were handling the specimens, and their DNA was sequenced. And it turns out that's a big problem. There was a paper published in 2009 or 2010 where researchers actually went into all of these non-primate genomic assemblies and DNA databases, and many of them had up to 10% human DNA contamination in them. In fact, there was an entire region of the zebrafish genome that was fully human because it was human DNA contamination that was assembled into the zebrafish genome assembly.
Oh, dear.
Yeah. And so what does that tell us also about all these primate genomes that are being sequenced? How much of them is just really human DNA and not ape DNA? And of course, it's really bad in these Neanderthal and Denisovan genomes. You just can't tell the difference. These are archaic humans. How do you tell what the difference is between a lab worker's DNA and what you're getting that's actually real archaic DNA? It's just nearly impossible to tell. There are different ways that they use to try and filter that contamination out, and they claim now that they can get only 2 to 5% contamination in a modern new genome assembly, but I'm pretty skeptical about that. We know how fast DNA decays. We know how fast it decays under certain temperatures, under certain moisture conditions, and it's just literally impossible for these remains to produce viable DNA after more than about 5,000 years, 5 to 7,000 years, 10,000 max under really good conditions.
And so that would probably be a pretty safe date to place upon, say, the Denisovan and Neanderthal skeletons, simply because we can extract DNA from them. And it decays so fast, it's pretty solid to say that they can't be any more than 10,000 years old.
That's pretty safe.
All right. Okay, well, thanks so much for taking the time to be on the show today. I know the viewers appreciate it.
All right, thank you.
Dr. Tomkins and ICR have an excellent new video series out entitled Unlocking the Mysteries of Genesis, which of course includes lectures from Dr. Tomkins and Brian Thomas, as well as many others. You can get your own copy at unlockingthemysteriesofgenesis.org. This interview was heavily edited for television. You can catch the entire meaty interview on our YouTube channel. Just head on over to genesisweek.com, which will take you to our YouTube channel, and you can watch the entire interview, which was pushing 40 minutes long. You can also subscribe to our YouTube channel while you're there. Stick around. We'll be back in just 1 minute.
Oh, not again!
To the horror of both fans and enemies, Ian Juby is back with more ranting goodness.
Okay, Jacques. You first.
Just when you thought his meds had kicked in, Ian goes off on a tangent about what killed the dinosaurs, the origin of life, defining evolution, and yes, even sex. It wasn't enough for an R rating, but nowadays, what is? Volume 4 of his ever-popular and ever-hated Creo Rants has 8 new short, fast, funny, and hard-hitting episodes. You can get your copy on the soon-to-be-extinct DVD for $15 plus shipping and handling, or purchase the instant digital download of all 8 tracks for just $8. Or you can buy all 4 volumes of his world-infamous rants for the price of 3. Order your copies today and have a party with, like, popcorn and stuff. Visit Ian's bookstore today. Woohoo! Mail for me? After what was probably the most provocative show I ever produced, I was flooded with positive comments. Okay, I normally love your stuff, but this one takes it to a whole new level. What you said, comparing evolutionism to creation, is spot on and so precisely what I've been saying since my conversion from an evolutionist 15 years ago, that you could have been reading an old post of mine. Love it. I personally feel that this was one of your strongest episodes, Ian. The script was sharp, the comedy was great as always, but most striking was the presentation, a really high standard of profession. Praise the Lord, and may this show never end. God bless. All right, I gotta go. I'm your host, Ian Juby, signing off from this incredibly cramped show. Remember, you can send in your comments, questions, and hate mail to us in a number of ways. You can email us at comments@genesisweek.com, or you can send us a tweet at GenesisWeek, or you can head on over to genesisweek.com, which takes you to our YouTube channel. Find the most recent show and post a comment there, or you can leave a comment on our Facebook page at facebook.com/GenesisWeekTV. Remember those words of warning from our Creator and Savior, Jesus Christ, who said, I am the way, the truth, and the life. No man comes to the Father except through me. We'll see you next week.
We are a viewer-supported program and need your support to keep this program on the air. Please pray for us, and if you wish to financially support the program, Canadians can make a tax-deductible donation to Core Ottawa, Canada North Post Office Box 72075, Ottawa, Ontario, K2K 2P4. While we cannot offer tax-deductible receipts outside of Canada, donors wishing to financially support the program can do so online at ianjuby.org/donations, and thank you for your support.